Okay, so in this video we're going to talk about postsynaptic potentials. Now, a postsynaptic potential is the word we use to describe graded potentials in postsynaptic cells, like a postsynaptic neuron. Earlier we talked about how neurotransmitter, once it's released from your presynaptic neuron, it can bind to receptors on the postsynaptic cell's membrane, and if it's a ligand-gated ion channel, this can cause graded potentials to form. Now, these graded potentials are generated as a result of changes in permeability of ions, and therefore a change in current, which changes your voltage. And the amount of neurotransmitter that's released can alter how large these graded potentials are, and how long that neurotransmitter stays in the synaptic cleft also will alter how large these graded potentials are. And an example of this would be, let's say if your presynaptic neuron released a tremendous amount of neurotransmitter into the synaptic cleft. Well, you're going to have even more neurotransmitter available to bind to receptors, so you get even more current in the postsynaptic cell, which means you even get a larger graded potential. And or we can talk about how if neurotransmitter stays in the cleft longer, you're going to get the same effect. So these are both modifiable in a variety of ways. Now, graded potentials are not always depolarizing. In fact, graded potentials can either be excitatory or inhibitory. So these excitatory postsynaptic potentials are a type of graded potential that's due to a depolarizing current within our postsynaptic cells. Now, the inhibitory postsynaptic potentials are actually caused by a hyperpolarizing current within our postsynaptic cells. So neurotransmitter binding opens chemically gated ion channels. And in the case of excitatory postsynaptic potentials, this allows for the flow of both sodium and potassium. However, because of the structure of these channels, sodium influx is actually much greater than potassium. And so because you're bringing in more positive charge than positive charge might be leaving, this influx of positively charged sodium causes excitation of the cell because it's depolarizing. And if you depolarize the voltage of that cell, you get closer to action potential threshold, and in that regard, it's actually excitatory because it might trigger the action potential. Now, what happens is these excitatory postsynaptic potentials are graded potentials, and so therefore they degrade over time and distance. So an IPSP has to be large enough so that when it spreads throughout the neuron, and by the time it gets to the axon hillock, if it's above action potential threshold, that can trigger opening of voltage-gated sodium channels, which then causes the action potential to be generated. So what this slide is showing is basically an example of an IPSP, where we have membrane voltage or potential on the y and time on the x-axis. So you see that in response to a stimulus, like neurotransmitter binding to receptors, you get an influx of sodium into the cell, which causes the voltage become more positive. But this is temporary because that sodium dissipates, and that its local effect on the voltage becomes smaller over time and distance. And so in that regard, graded potentials are these temporary changes in the voltage of our cells. Now, if they're excitatory postsynaptic potentials, they allow for sodium to rush into the cell and are therefore depolarizing. And it's excitatory because it gets you closer to action potential threshold. Now, in this example, an action potential wasn't generated because the graded potential, or IPSP here, wasn't quite large enough to get to threshold. Now, opposite of that, we have IPSPs, or inhibitory postsynaptic potentials. So these inhibitory postsynaptic potentials or IPSPs, are hyperpolarizing, which means that due to the current that they generate, it causes the voltage in the cell to become more negative than rest. And that's what hyperpolarization is. Now, this hyperpolarization of an IPSP is due to either potassium or chloride flowing. Now, potassium flows out of the cell due to its electrochemical gradient if it flows through a certain channel, and then chloride can flow into the cell due to its electrochemical gradient if it's allowed to. So what we're saying here is that if a neurotransmitter from, let's say, an inhibitory neuron binds to a certain receptor on the postsynaptic cell, it might allow for potassium to flow out of the cell or for chloride to flow in, and both of those types of current causes the cell to become more hyperpolarized, which is essentially when the voltage becomes more negative than rest. And this is inhibitory because it gets you farther away from action potential threshold. So to look at what an IPSP looks like on a voltage trace, we see that we have our stimulus here, and that in response to, let's say, neurotransmitter binding to its receptors, and all of a sudden we get potassium leaving the cell, if all of a sudden you make potassium even more permeable, it's going to flow out of your neuron and make the voltage even more negative because you're removing positively charged ions. Same thing with chloride, except for chloride could be flowing into the cell, and because you're bringing in negative charge, that also makes the voltage more negative. But remember, this is a gradient potential, so it degrades over time and distance. And it's inhibitory because it gets you farther from action potential threshold. Now, what's pretty interesting here is that IPSPs and EPSPs can summate or add together. So you might have a depolarizing EPSP that adds together at the same time with a hyperpolarizing IPSP, and their effect could cancel out. Or you might have multiple EPSPs add together to get you to action potential threshold. So most of the neurons in your brain, spinal cord, and around your body receive both excitatory and inhibitory inputs from thousands of other neurons. So basically, in order to get to action potential threshold, we typically need several EPSPs to summate. That way we can generate an action potential. Now there's two types of summations here. We have temporal and spatial. Now temporal summation occurs from typically about one synapse, and if this one synapse transmits action potential, generates depolarizing or hyperpolarizing a current very frequently, such that those gradient potentials add together in time, then they can summate, and then we call this temporal summation because it occurs typically at one synapse. Now, spatial summation is when you have a postsynaptic neuron that's stimulated by a large number of synapses simultaneously. And you might find that there's thousands of EPSPs and IPSPs that can add together and summate to reach some kind of net effect. You know, you might have 5,002 EPSPs and 4,015 IPSPs, but because of the fact that you have a lot more EPSPs and IPSPs being generated simultaneously, that means the net effect on that postsynaptic neuron is excitatory, and that could generate action potentials in that neuron. So this is actually a pretty cool way that neurons can actually either excite or inhibit other neurons. So what this is showing is an example of where we don't get integration or summation. So you might have a circumstance where one neuron excites a postsynaptic neuron here, and because the excitatory events or stimuli occur too far apart in time, the gradient potentials that are generated in the postsynaptic neuron don't add together. In fact, they stay separate, which means you actually don't get to action potential threshold. But if you added these stimuli closer together, summation would occur, and because these EPSPs could add together, it could get you above action potential threshold. And that's what we see here. So if we actually have this one neuron generate action potentials more frequently, and therefore excite the postsynaptic cell more frequently, then we see here that these stimulatory events are closer together, so that the first EPSP can add with the second, and because that gets you to action potential threshold, then we get an action potential. So then you get the depolarization phase and the repolarization phase of the action potential, and that's going to spread throughout the axon. Now this is an example of temporal summation because it occurs at one synapse. Now spatial summation would occur at many synapses, and this could be like where we have an EPSP from one synapse add together with an EPSP of another, and because they add together, then they can get you to action potential threshold, which then generates your action potential. Now what's interesting too is we can also throw in an inhibitory effect here as well. So let's say that we have an inhibitory or IPSP gradient potential generate because of an influx of chloride or an efflux of potassium, that it hyperpolarizes the voltage and takes you farther away from action potential threshold. Now, because this one event, occurred way back here, it basically just made it less likely for action potentials to be generated. But let's say if we have an excitatory and inhibitory gradient potential, basically add together or summate at the same time, they almost cancel each other out, where you just see the tiniest little blip or change in voltage here because of these gradient potentials adding together. And so because these add together in time, you can prevent this neuron from responding. In fact, a lot of inhibitory types of synapses are actually located closer to the axon. That way they can block the generation of an action potential and be a better inhibitory type of synapse. Now, synaptic potentiation is a really interesting effect of neurons, where repeated use of a synapse increases the postsynaptic cell's ability to respond to that synapse. So think of synaptic potentiation as like positive feedback, where once you generate a response in the postsynaptic cell, that response makes it far more likely that the next response will be easier to generate. And so the way this works is that by repeated excitation or inhibition of a postsynaptic cell, calcium concentration can increase in the presynaptic terminal, which causes the release of more neurotransmitter, and this leads to more IPSPs in your postsynaptic neuron. Also, what happens here too is that potentiation can actually cause these voltage gates to open on the postsynaptic neuron, and calcium in the postsynaptic neuron can activate these kinase enzymes, and these kinases can basically alter the sensitivity of receptors on the postsynaptic cell, and therefore make them more likely to respond with even less neurotransmitter. So what's cool here is that this is actually the chemical correlate of learning. In fact, we call this long-term potentiation, or LTP. So long-term potentiation is how synapses can strengthen over time from repeated use. And this is why it's important that if you're learning something for the first time, that you have repeated repetition of that over and over and over again. That way you can stimulate long-term potentiation, the formation of newer, stronger synapses that can help with learning and memory. Now, what can also occur is presynaptic inhibition. And inhibition is actually due to the release of excitatory neurotransmitter by one neuron, but can be inhibited by another neuron through an axo-axonal synapse. So what's pretty cool here is that we can see then is that you can block the excitatory effect of one neuron by causing inhibition by another. And so what we see then is just basically a summary of where graded potentials can occur. Remember, graded potential occurs in the cell body and dendrites, and those can spread on over to axons. Now, remember, graded potentials typically travel a pretty short distance, and they get smaller over time and distance as they travel. But if these graded potentials have enough depolarization, by the time they get to the axon hillock, they're going to generate an action potential which travels long distance and very rapidly. In fact, much more rapid than a graded potential. Now, in terms of a graded potential versus an action potential, just a comparison and contrast, we see that the amplitude can vary in size, and that's what makes these graded. However, action potentials are always the same size, and they're all or none. And graded potentials decay over time and distance, whereas action potentials don't. Now, graded potentials are stimulated by lots of different types of stimuli. It could be light, pressure, temperature, and action potentials are only triggered by depolarization. So you need enough depolarization at the axon hillock to get to action potential threshold for these to be generated. Now, we see that graded potentials don't typically respond to positive feedback types of regulation, whereas action potentials can. In fact, that's how action potentials work, is that due to opening of voltage-gated sodium channels at threshold, it causes even more voltage-gated sodium channels to open, and that's what triggers the depolarizing phase of the action potential. We don't see this in graded potentials. In fact, graded potentials don't stimulate themselves, which means they don't propagate and therefore degrade over time and distance. But because action potentials have a positive feedback cycle, they can propagate themselves and travel a long distance. Now, repolarization of graded potentials occurs when no stimulus is present, because these things get smaller over time and distance because of diffusion. Action potentials repolarize when the voltage-gated sodium channels close and the voltage-gated potassium channels open. So you need these voltage-gated potassium channels to repolarize the action potential. We see that graded potentials occur due to a stimulus from other neurons at a synapse, and it can either occur either temporally or spatially. Now, graded potentials occur in two major types. We have excitatory and inhibitory postsynaptic potentials. Remember, excitatory postsynaptic potentials generate depolarizing current, which is typically sodium flowing into the cell, and inhibitory postsynaptic potentials, or IPSPs, generate hyperpolarizing current, and which is typically potassium efflux or chloride influx. What we find, though, is that action potentials only have one form. There's not like inhibitory or excitatory action potentials. These things just travel long distance. They constitute the nerve impulse. They're the same way every time, so long as everything is working properly. Now, we find that gradient potentials are caused by ions flowing through different channels. So remember, the IPSPs are due to sodium influx, and because of that net influx of sodium, you're bringing more positive charge inside the cell, which makes your voltage slightly more positive. And that these IPSPs are generated by potassium efflux or chloride influx, which is what hyperpolarizes the voltage away from rest. Now, action potentials are caused by two types of voltage-gated channels. Voltage-gated sodium, which is what constitutes the rising phase of the action potential, and voltage-gated potassium, which is what constitutes the repolarizing or falling phase of the action potential.